individual views, criticism and review. So you've got to criticize them, review them. It's a good idea. All right, we're going to be talking about circulation of the cardiovascular system. This is usually a very, very long topic. The only system we cover in this term. The question is why you have the requirement for a circulatory system is an evolutionary consequence of the increasing complexity of large multicellular organisms, which we are one. Diffusion is not adequate, the distances are too large. So if you have, so this diffusion is too slow. Diffusion of all sorts of things would take its proportion to the square root of the distance, the time. So if it's a centimeter, by the time you've got some sort of oxygen or something to a cell, it might take a few days. So the primary role for the circulation we're going to talk about, and we're not going to spend much time on it, is to provide nutritive elements to cell, all cells. And eliminate waste products. So you have a circulation, and that's primarily its function. Primary function. If you were, I hope not, a large unicellular organism, then this would be the situation. Some amoeba or something grew out of a sewer in Granbury, Alberta, sort of really deep down. Not too far. So basically, you have a membrane from this multicellular or a single cell organism. And you have driving forces for nutrients and another driving force for waste products across the plasma membrane. So that would work, but if you only one for two cells. However, we're more complex than that, so we need circulation. So the circulation basically provides a steep concentration gradient to close proximity to every cell in the body, human, so that every cell is within 10 or less micrometers from the supply of blood. And from the blood to cells, you have nutrients, which could be oxygen, glucose, vitamins. And from the cells to blood, waste products, carbon dioxide, urea, lactate, all sorts of things like that. So basically, you have a steep concentration gradient provided by the circulation in very close proximity to every cell. Okay, that's the primary function of the circulation. There are several secondary roles for the circulation as well. The first one is fast chemical transmission of information, and then an example of that is circulating hormones. We all sort of secrete hormones that are secreted at one location and travel through the blood to another location. Dr. Barton will be giving you one example, and what would that be? Something is secreted in one location and goes through the circulation to all kinds of other sites. I'm sorry? Yeah, what about, if you heard of the adrenal medulla? That's a big one, right? Fight or flight. So basically it secretes a mixture mostly of adrenaline. The adrenal medulla is located just over each kidney. You have gland that secretes adrenaline, noradrenaline into the bloodstream, mostly adrenaline, and it's into the circulation and it travels and it does all sorts of things all over the body, including the heart. Okay, so that's what one aspect of the circulation. Dissipation of heat, or retention of heat. Saturday with nice wind chill out there. You went outside without your gloves, and your hands turned white or pale. The reason for that was, of course, is the divisions of the autonomic nervous system constrict blood vessels to try and reduce the loss of body heat. On the other hand, you head down to Houston, Texas, and it's, I don't know, 45 outside in the shade, humid, and of course you get your hands within, after a couple hours, will turn a little darker color. In fact, you're getting increasing circulation to the skin to remove body heat. Okay, so that's another aspect of the circulation, secondary road. And of course, it travels, macrophages and things, white blood cells travel to various locations in case there's an infection. So it's a target infection. So mediating inflammatory and host defense response to hasten, creating microorganisms. You get a cut, start to get infected, once you get a little red in that region, to the circulation. So those are secondary roles for the circulation. The primary ones, of course, is to provide nutrients such as oxygen and remove carbon dioxide from the cells during the metabolic process. Okay, so what is the circulation? This is something here, it's a bit of an issue. It's thing has to be adjusted. We'll get that done. The system of circulating fluid is driven by a pump. So circulating system includes the heart, the blood vessels, and of course blood. This involves blood. So the system of circulating fluid, which is blood, is driven by a pump called the heart. Now, a lot of you country and western fans know that major hits say that the heart is the seat of love. She done broke my heart, or my heart is broken because my horse left me. Or it's broken because my girlfriend came back. It's broken anyway. The issue is, of course, is that you're going to get some lectures on endocrinology and reproduction later on in 214, which is probably more relevant than, of course, the heart. But the heart is basically a very unique, and in my opinion, a very unique pump. Unbelievable, actually. Anyway, so this pump, called the heart, pumps about 5 liters of blood, depending on the size of the individual. If you're Patrick Kabongo, it's probably 6 liters. If you're someone like me, about mine's about 5. So it depends, size. But that's usually the sort of numbers that are given per minute through a series of distributing and collecting tubes. An extensive system of thin-walled vessels. We're going to talk about all of these things that permit rapid exchange between the tissue and the vascular channels. So basically, you want to get nutrients in and out of cells, nutrients and waste products in, nutrients in and waste products out of cells. Okay, and these blood vessels, or this plumbing, consists of something called arteries. Of course, you all know that. Arterioles, capillaries, where all the action occurs, and then the return part, venules and veins, which comes back to the pump. This is a simple example of part of the circulation. It eliminates the lungs. It only looks at the systemic part where the blood is fairly high in oxygen. And it consists of the arteries, which are large tubes, which terminate in something called a peripheral resistance, if you're looking at pressure regulation. And in this region are arterioles, small vessels, and most importantly, the capillaries make up what is called a peripheral resistance. So this is a simple model, and the capillaries are where all the exchange occurs. And of course, the blood then comes back to the veins, going into actually the right atrium, gets oxygen, and comes back around. So that's a simple schematic of circulation. Talk about the heart. Well, the heart, we spent some time on the anatomy, largely to get the nomenclature down. For no other reason, because I'm sure that all of you, after taking biology 20 or 30 or something, would have dissected a beef heart or something. I would assume. How many have? Gosh, you can teach me stuff I never did. Okay, a human heart consists of 4 chambers. Unlike some of the things like frogs, which people work on experimentally from time to time, only have 3. 4 chambers. Has 2 atria, a left and a right. They're thin-walled structures, relatively speaking, about 1 to 2 millimeters thick. And it has 2 ventricles, also a right and a left. The atria basically are thin-walled, low-pressure chambers. And although they do contract, their function is mainly to act as reservoir conduits that pool blood. Conduits of blood to the respective ventricles. So that's their primary function. They do contract, but the contraction is, in terms of getting blood through that circulation, is irrelevant. It doesn't really make any sense. It doesn't do anything. They store blood, or basically pool blood, so that if you have blood, when the valves open, blood floods into the ventricle. So that's their primary function. The ventricles, on the other hand, are where the action is. That's a real pump. And they're responsible for the forward propulsion of blood. The right ventricle develops a mean pressure of about 1/7 that is developed by the left. So there's a low pressure side, which is the right side, and a high pressure side, which is the left side. And in keeping with this, the right ventricle is quite a bit thinner wall thickness than the left, about 1/4. Okay, so that's roughly sort of the basic structure of the heart. Here's the diagram. This is actually all the stuff from Netter, the medical artist, about 35 years ago. He could draw all these things very well, and that's one of his. And this is just a schematic representation. Right atrium, right ventricle, left ventricle, left atrium. Now, just notice here, the left side, as I initially pointed out, is quite a bit thicker than the left side is quite a bit thicker than the right side. So that's sort of in keeping with what I said. And that's because it's developed higher pressure. It needs more muscle to do so. Now, in order to have any kind of pump and do anything, you've got to have something in and out. So if it's pumping out the blood basement, you've got a tube that sucks the water out and then goes through the pump out to the lung. And of course, the circulation has various blood vessels. There's something called a pulmonary artery, and that goes from the right ventricle to the lungs. It comes back from the lungs through pulmonary veins back to the left side. And that's called pulmonary veins. And in the human, there's usually 4. Sometimes there's only 3, but in general, 99% of the people always have 4 pulmonary veins. The major high-pressure blood vessel is the aorta. It goes from the left ventricle, carries blood to all organ systems except lungs. And that's strictly not 100% true. So kind of pick up our shunts. We'll bring those up later. The inferior vena cava provides venous return from the lower parts of the body. That's most from the neck down, more or less. And the superior vena cava, the venous return from the upper parts of the body. That's why it's called superior, because it drains the brain. Here's a diagram showing you the major blood vessels through, this is also artistic representation, but it's fairly close. Ventricles. This is the aorta, the arch of the aorta. And this is pulmonary artery, goes to both sides, right and left. The venous return comes back to the left atrium through 4 veins. And of course, the biggie, or the most important venous return, is the superior vena cava, and this is the one that comes back from the lower parts of the body, the inferior vena cava. So that's a view of all the blood vessels. What is the heart made up of? The muscular tissue is what we're talking about briefly, cardiac muscle. % of the number of cells, if you actually want to go through, take somebody's heart after they're dead and count different kinds of cells. I don't know why anybody would want to do that, but somebody apparently has, roughly. 30% of the cells are muscle, the rest are fibroblasts. However, when you look at the muscle mass, 70% of the weight is due to muscle of the heart, and 25% various kinds of fibrous tissue, the vessels, fibrous tissue around the valves, etc. So most of the mass of the heart is made up of muscle. Now let me state this before we go on, or reinstate it for you, is that I did say cardiac muscles are type, but depending on the function, all cardiac muscle cells are slightly different, or different. Some quite different, some just slightly different. So they're not all the same. It depends on their function. Okay, so that's the blood vessels. We have epithelial tissue, inner covering of the heart, so it gives it a protective lining over the muscle. And there's also something called a pericardium, which is a fibrous sac which covers the heart unless you've had open heart surgery. So it's there. And unfortunately, if you get the odd viral infection, which cause fluid to build up between this pericardium and the actual heart, so then you have severe chest pain, gives you strange ECG recordings, etc., etc. So basically, and then you actually get fluid buildup. So your friendly neighborhood cardiologist puts in a needle, a syringe, and sucks out the fluid, gives you some antibiotics, and that's the end of it. Anyway, that's the pericardium. It's a fibrous sac which covers the heart. So the other type of tissue is supporting tissue, connecting tissue between the atrium, the ventricle, and valves. And then some of the valves have what are known as fibrous valve rings. So you have, in order to have any kind of pump, you got to have valves so that the fluid, in this case blood, flows in the right direction. Otherwise, no valves, no pumping action. So you got to have valves. The valves are usually thin flaps of flexible, tough, endothelium-covered fibrous tissue attached at the base of the valve rings in some cases. Fibrous tissue of valves is collagen, and the valve rings are usually cartilage. So that's a valve, you have valves. This is the pump. And it's 2 pumps in series. Okay, now valves. What are these valves? Now, physiology textbooks, I'll like to refer to 2 sets of valves, or have called the atrioventricular valve. And these are the valves between the atria and the ventricle. Now, in general, you go to your friendly neighborhood cardiologist and tell them there's something wrong with your left atrioventricular valve. I'm not really sure what you're talking about because they've actually been given medical aid. So basically, the right atrioventricular valve is known as the tricuspid. And that's what you'll see on exams. Consists of 3 leaflets or cusps joined at the circumference by a fibrous ring made of cartilage. The other one, the left atrioventricular on the high pressure side, consists of 2 leaflets attached to a fibrous ring, and it's called a mitral valve. So you should remember. These valves are part of an apparatus, and they have something called chordae tendineae, and it strengthens the valve during contraction of the heart and prevents the opposing borders of the valve from inverting. And chordae tendineae is very fibrous tissue, and anybody has done any baling or haying, you see that it looks sort of like bale or twine. It's very coarse looking things. And if you've dissected beef heart, you know what I'm talking about. Anyway, that's their chordae tendineae, and that's their function. And they're connected to something called the pillary muscle. This is continuous with the ventricle. So contraction of the pillary muscle prevents reversion of the AV valves during contraction of the ventricle. So keeps them in the right orientation so they don't invert. Here's another representation. This is the mitral valve. See the valve leaflets? These little projections here from the ventricle are papillary muscle. And these guys here, these sort of looks like threads, are in fact chordae tendineae. So basically when the ventricle contracts, these things shorten and make sure that these valve leaflets don't go backwards. It gives you good closure. So that's a look at one of the atrioventricular valves, in this case the mitral. It consists of this entire apparatus. And there's only 2 valve leaflets and there's a pillary muscle controlling it. So these things, when the ventricle actually contracts, these things actually shorten. And they sort of maintain some tension on that, so the valve leaflets close like you're praying. Now, in some cases, the valve leaflets are redundant. They become very large, and that's genetic malformation. So and it actually can bend over and go into the atrium. That's called mitral valve prolapse. And in that case, unless they're very large, it's not really a big problem. But if they're very large and then they leak, then of course you need some surgery to replace your mitral valve or to strengthen it or take out some of the excess. So these are usually normal and close fairly well, and they of course are located on the high pressure side. The arterial valves, it's called semilunar valves. Somebody initially, when they dissect the heart, said they look like a half moon. They're pretty well similar in both cases. Each valve consists of 3 pocket-like cusps of approximately equal size. Just like, I don't know, somebody said it. Things you hold pies in Tupperware or something, told me once. All the guys know what I'm talking about. Anyway, equal size, and there's no distinguishing fibrous tissue. There is fibrous tissue in this region, but it's not distinct. The pulmonic valve is in the pulmonary arteries on the low pressure side. The aortic valve is in the high pressure side. And these are the 3 valves. I'll show you a diagram. These both, the aortic valve in particular and the mitral valve on the high pressure side, are susceptible to infections by what is known as rheumatic fever. And you get all kinds of strange things. For example, the major one is a non-autoimmune condition caused by a streptococcal infection. And valve leaflets, the mitral and the aortic, become damaged. They sort of get little pimples on them, pustules, that's due to not being the new condition. And that was very common years ago before penicillin hit the market. And it's still around, I guess. And in fact, if you've had, somebody's had rheumatic fever, and you go to your dentist, the usual thing is before you get your teeth cleaned or any kind of dental procedure, you have to go on a 1 week or so tour of using antibiotics to prevent reinfection. But reinfection makes these things bigger and bigger and bigger, and these valve medicine become damaged and have to be replaced. So it's not as common as it used to be, but it's still not too many years ago. Somebody came up after class and said, well, I'm on antibiotics before I go see the dentist. Why is that? And that's the reason. You don't want a reinfection. This is again a diagram. It's kind of hard to get this diagram by doing dissection. This is a representation showing the triflecta, mitral, and the two pulmonary valve, the pulmonic valve, and the aortic valve. So these are the pocket-like cusps. The AV valves have this little ring of cartilage around them. And these are cartilage or valve rings, virus valve rings. These have some cartilage in there, but it's not as distinct. And that shows the side view. This is the aortic valve. Valve is there. This is a chunk of papillary muscle, which is shown the cut, and both have papillary muscle, and these are in fact the valve to that kind of funny shape. The other thing I want to point out, we're talking about atria being one kind of chamber, and it is. However, the little pouches on top of the atria called auricles. They don't do much, except for one specific case. When the atria is not functioning properly, blood pools in there. Especially in older individuals who get atrial fibrillation, the blood will actually pool in there. And what did Dr. Barton tell you about blood clotting? It becomes static. And the thing is, once it becomes static in these little pouches, it sometimes clots, goes through the circulation, lodges in your brain somewhere, and you have a little trouble remembering your name, you get a stroke. So that's one of the issues there. The other thing, if you do bypass surgery of the heart, that's a good place to stick cannula. So these are the little auricles, there's one on each side, little pouches on both sides of the chamber, the right one and the left one. But they're continuous with the atria. The only thing is they are pouches, and they could actually collect blood, and if it's not pumping properly, fluid, and you get clotting sometimes, you get a clot and it goes through circulation, you're in big trouble. This is just a diagram showing rheumatic fever, and you just get these little bits or little pimples on the valve underneath us. This is the mitral, and this, of course, is the aortic valve. That's what I'm talking about, rheumatic fever. Right? You get little growths on there, and this is not a big deal. However, if you have several reinfections, go through this stage here where they actually become a problem. Valves don't close properly. So you get either valves replaced, either or ball and cage, but they get really bad, and then of course they don't open it. There's insufficient, there's actually blood goes back and you can't pump blood. properly. So that's a consequence of reinfection of rheumatic fever. Okay, so basically this is the heart. Gross anatomy, I'm sure that all of you this is all over the path for you. Histology of cardiac muscle. One of the things I'm going to come back to probably at the end of the lecture, there's a spiral arrangement. And that is important in how the left ventricle contracts. And to sort of repeat what we talked about muscle, heart muscle consists of a network of cells which are joined longitudinally and anastomose and neighboring cells by short bridges. Adjacent cells are held firmly together by an elaborate zigzag intercalated attachment. It's called an intercalated disc. You've seen this diagram before. Shows you the three-dimensional structure of cardiac muscle made up of small cells. This is more or less the ventricle, and of course you have these little regions of attachment called intercalated discs. Here's one, here's one. Those individual cells, looked at these before, so they're not very large, about 15 micrometers in diameter, in the order of 100 micrometers in length. In this diagram, this is of course an electron micrograph of the section of the ventricle, and it shows you this intercalated disc. This region has several structures. So this is a schematic representation. There's one here, there's one here, one here. So these two some sort of desmosomes, and they provide mechanical coupling to the heart. So one cell contracts and pull tugs on the other. And this sort of region of attachment is where you have gap junction. So these guys provide mechanical coupling to the cells. And these, of course, do something else. They couple with heart cells electrically. So if you look at that little region, number one, the spot desmosome. The next one's a belt. And you have nexus or gap junction. These two guys here, 1 and 2, provide mechanical coupling. So the heart, all the heart cells are fairly tightly mechanically coupled. Because they're small cells, and they're all individual guys, but they have to be mechanically coupled for chambers to contract. The nexus or gap junction provide electrical coupling. So the gap junction, the surface membranes of two cells are very closely opposed. So 20 angstroms, 2 nanometers are bridged by tiny stalks, 2 nanometers in diameter, and you have a tunnel diameter of about 1 nanometer, which is very large. This carries ions, and of course various metabolites between cells. The gap junctions at the intercalated disc are responsible for the spread of action potentials over the whole chamber, either the atrium or the ventricle. The 8-cell is like so. In summary, although the heart is made of individual small cells, it is tightly coupled mechanically through desmosomes, tie the cells together mechanically, and it is tied together electrically by gap junction. Electrical synapses. So it's got great electrical coupling, and it's got great mechanical coupling between cells. It has to have. This is one of the desmosomes. I missed something here. But anyway, these things are stick together, drop calcium, a few enzymes, and you can separate cells. That's what we did not the diagram I'm showing you, but we've done that sort of thing with heart muscle in various species. And we couple the actual cells to each other. These are, of course, the plasma membranes of the individual cells. The other one, of course, so these are the mechanical coupling. That's the important aspect. The other one is, of course, the electrical coupling. This is a region showing the gap junction. This is one cell, this is the other cell. These are all called connexins, there's 6 of them. And they couple the cells electrically. These things are sensitive to all kinds of things. Calcium, protons. So everything's working normally and the pH is around 7.2 or whatever in the cell. These things are open. If the cell loses oxygen, as you might have a myocardial infarct, and one of the things that happens, the cells start to die, or some cells start to die. And when that happens, the immediate thing that occurs, of course, the pH will change. More protons in there, and the other thing that occurs, of course, you get calcium flooding into the cell. If that happens, these things then rotate and close off completely. So they basically uncouple electrically and metabolically the live tissue from the dead tissue. So that's the point of the fact that it opens or closes. Normally they're open, but if you increase calcium inside past sort of normal rate for a period of time, or change pH by having proton, these things will rotate and close off. So that's an uncoupling process, sometimes referred to as healing over. So basically, you've had a minor heart attack. Within a few days, the dead tissue is uncoupled electrically and metabolically from the live healthy tissue. So that's a function of these guys. But in summary, cardiac muscle cells are small, make up the chambers in 3 dimensions. They are coupled mechanically through desmosomes and electrically through gap junction. So the heart acts as one, chambers act as one. Just another point. The only other thing I want to mention is I talked about the spiral arrangement. The right ventricle, when it contracts, is simply a change in diameter. So if the diameter is, the radius is 2 centimeters, contraction will go down to 1.5. That's the way the right ventricle contracts. The left ventricle, on the other hand, has coupling in preferential, mechanical coupling in preferential directions. And this is sort of somebody's artistic representation of that might be. So when the left ventricle contracts, there's the preferential coupling, mechanical coupling. It not only contracts and changes the diameter, but it also rotates a little bit. It sort of squeezes the blood out like you're wringing out of the washcloth. So it's a little different. The right side is just simply a change in the radius. The left side, of course, in addition to that, does a bit of a squeezing effect because of the spiral arrangement of the mechanical coupling. That's what I mean by spiral arrangement. So it's only the left side. So mammalian cardiac cells are coupled by a small number of gap junctions. Cells communicate with each other, and this arrangement is called a syncytium, meeting together cell. And the idea that sort of focused or pointed out in many textbooks, certainly the old one, heart is a functional syncytium, so if one cell is excited, the excitation spreads over the entire ventricular arrangement. That's kind of a lie. because we need to activate about 10 to 15 cells. The currents aren't enough out of one cell to generate any kind of propagation. So you need several cells, more than one. But this is the old hypothesis. So one cell is excited, everything's excited, not likely. Current spreads in 3 dimensions, and we have to activate a cluster of cells. And it basically says if you get this activated, you use the whole chamber, for example, the law of non-entropy. So if you activate a part of it, it'll spread to the entire chamber. Oh, this is bad, okay, this is a one-dimensional representation, but we have a three-dimensional representation showing you how cells are coupled. The gap junction of this looks like, if this was the case, this would look like a nerve cell. Where you have low resistance pathway, you have intercellular current, which will be polarized, and extracellular current, which will be polarized adjacent areas. This is all like a nerve action, unmyelinated. Question I have, so you have intercellular current carried by ions. Go back to Dr. Noyen's lecture, what could be the major current carrying ion inside? What choice have you got? So what's the highest concentration of what ion inside cells? Potassium. That's right. So carried by potassium largely. And of course they carry other things. They'll carry calcium ions. Although normally, unless it's contracting, there aren't many. They carry metabolites, as the cyclic AMP would actually go through there. And of course, this is one-dimensional, right? Because you would also have, in a real heart, you would also have connections in cells in 3 dimensions. So this is a one-dimensional intercellular current going through the gap junction to polarizing adjacent cells. Okay, well this is, we've done this before, been there. Similar to skeletal muscle, shows banding, cross, longitudinal, has A band, I band, Z line, H zone, M line. Same thing as in skeletal muscle. The important guy here is, of course, in ventricle particularly, is the protein titin, which gives you some elastic recoil to the ventricle through the contraction of the muscle fiber, or muscle cell. So that's important. Tight. And of course, membrane is an area attached to the deadline. Cardiac muscle has a T system and an SR. SR is not as well developed as in the skeletal muscle, but it is an important component of EC coupling, as we talked about a day or so ago. This is in fact another representation. Nebula in there, and this is titan, providing the elastic recoil. And I said if you stretch this too much, of course you will get shrinkage of those and you get all kinds of issues with contracting cardiac muscle cells. So I'm going to hit through this again. Cardiac muscle cells are smaller than skeletal muscle cells, 50 micrometers in diameter. These guys are 100. And the point I want to read is that the external calcium, which in you right now, unless you've had 17 glasses of milk, is about 1.8 millimolar. But this 1.8 millimolar outside the cells is crucial for EC coupling. That is not the case for skeletal muscle. It is the case for cardiac. You've got to have a certain calcium. So, as I've said before, cardiac muscle at rest. In a normal person, the ventricles contract about 60 to 80 times per minute. And if you live to 75 years, Don't party too much. Your heart can actually contract about 10 times 8, 3 times 10 to the 9th, or in a 75-year lifespan. Hopefully you'll live longer than that. But basically, it contracts a lot of times. That's a lot of repetitions. Try to do that with your TV remote. Anyways, it's a lot. So how is this controlled? And as I said before, the rhythmicity of the heart is myogenic in origin, myogenic or a muscular origin. So it does not require nerve action potential from any place to actually contract. Case in point, in heart transplants, everything is cut and the heart contracts. Now usually, in many cases, they implant or stick in an artificial pacemaker, but that's the, doesn't really matter. It'll contract at some rate. The highest rate occurs in a region close to the junction of the superior vena cava, which is one of the veins, with the right atrium. It is located in an area of about 2.5 centimeters to about 0.4 centimeters. This is the region. And this area is known as the SA, or the sinoatrial node, or the pacemaker. It has the highest rate of spontaneous depolarization. And we'll go through how this action potential works next day. In the heart. So in terms of spontaneous activity by itself, it has the highest rate. Every region in the heart is capable of generating some sort of rhythm. But there is a hierarchy. Pacemaker is the fastest. Regions in the atrium would be number 2. And regions in the ventricle that might want to pace would be number 3. So the pacemaker overdrives all these other cells and suppresses them. That's called overdrive suppression. And I think I'm going to package it here. We'll start with how the pacemaker works next time.